friends, I am Ayantika and in this video I will be discussing about a general overview of structure of fungi. Now fungi or fungus, they are achlorophyllous organisms. They are thallophytic, which means that their body cannot be differentiated into a tissue organization or organ level organization. Apart from fungi, algae and bryophytes are also considered as thallophytes. So fungi, algae, bryophyte together they are considered thallophytes. And today in this video I will be discussing about the general structure of fungi. I have also made a video on the general features, characteristic feature of fungi, and I have put the link of that video in the description box. I have also made videos on other topic related to fungi as well as slime molds. Those videos also I have put in the link in the description box. So let's start with the video of the structure of fungi. So about the structure of the fungi, it is thalloid, which means that there is no tissue level differentiation. So it can be, the fungal can be either unicellular or it can be multicellular or huge. So this unicellular, they are generally in the shape of an amoeboid plasmodium, or they are circular, more or less circular. So this plasmodial form is found in the lower chytrids such as the Plasmodiophora and the Syncytium. Now the Syncytium, we know it is a very infectious disease producing fungi. It causes blackboard disease of potato. So the Plasmodiophora, Syncytium, these are unicellular amoeboid fungi, and in fungi which is more or less circular, like for example here it is the yeast, we see it is more or less circular. So this unicellular fungi, it is the entire cell. It accumulates food, it grows in size, and then after reaching certain stage of maturity, entire cell is converted to one or more reproductive units or reproductive spores, whether asexual or sexual spores. So this type of life cycle, it is called holocarpic, where this type of life form where the reproductive and the growth phase is does not occur in a single organism. So such type of life form is called holocarpic. In holocarpic organisms, the somatic phase is followed by the reproductive phase, and again the reproductive phase is followed by the somatic phase. Never do these two phases occur together. So after unicellular, we come to the filamentous fungi. Now filamentous fungi, they form a tubular structure that is called hypha. They can this tubular hypha can branch and ramify in various directions, and it can form huge structure. That is, it can be it looks like a cottony mass, and this hypha or hyphae which forms huge mass, it is this type of structure is called mycelia. So just a single hypha is hypha, but when it reaches enormous proportion, it is called mycelia. By enormous proportion, I mean that it is not as huge as, but it is macroscopic. So the hyphae or hypha, it is microscopic, but this mycelium can be seen with naked eyes. Especially in our MSc syllabus, we have this rhizopas and mucor. They form visible mycelium. Even we can see this penicillium, mycelium also on the surface of bread. So this type of life form is eucarpate, where the hypha, it ramifies in all direction, it branches, ramify and searches for food, accumulates food, and after certain time this say for example this is the substratum, and the hypha is growing inside the substratum, and then it will do is that it will give out aerial branches which are essentially the reproductive structure. Now this hypha, any filamentous hypha, they begin as spores. If the spores on reaching the suitable condition for growth, or if it is a parasitic fungi, then if it falls on the suitable plant, or if the spore is falls near the any growing plant, then it germinates by putting forth a germ tube, which is essentially a tubular hypha like structure, and then it grows into this type of structure. And after certain period of maturity, when it has accumulated a huge amount of food, then it sends out this aerial hypha. Aerial hypha is basically either sporangiospores or conidiospores. These are basically the reproductive structure. And this aerial hypha, it is colored. The aerial hypha, it can be orange, yellow, red, or even black or dark blue colored, or it can also be white colored, normal, translucent. But generally the reproductive structures, they are tinted, they are colored. So this hypha is also of two types. The hypha can be either aseptate, that is, it does not have septation, it branches but there is not septation, and a septation is found only when they reach the reproductive stage. That is, when they reach the reproductive stage, a septation is developed, and septa are basically cross wall. So when they reach the stage of maturity, they start to produce the reproductive organ, then only the septation is formed in the aseptate variety. Whereas in this, here we see the septate variety, septations are present right from the beginning. So in the there is two type of mycelium, septate and septate. We'll be discussing it more in the next slide. So this aseptate, they form septation only at the time of reproduction, and so they are cytic, and this type of mycelium is found in the lower fungi, that is the Fungomycetes. And we know that a collection of many hypha, when it reaches massive proportion, it is called mycelium. So mycelium is aseptate, and when it is septate, septations, they are formed. Here it is the example of septate mycelia, where we see that the transverse wall, it undergoes centripetal growth, that is, and forms a tubular structure. So for example, this is the hypha and this is the tubular wall. So growth will start centripetally from all direction, and then annular rim-like structure is formed. So this septa, it is not complete. Here we see that a pore is left behind. So this central pore, it is instrumental in developing the protoplasmic continuity, that is, the organelles as well as the food matter, it can travel through this septa in the through the hole in the septa, the central pore in the septa. So this is not a complete septation. Protoplasmic transport occurred, as well as transport of nutrient occur, and at the same time, if there is any information, chemical information that needed to be transported, transfer of chemical information also occur. Now, what do I mean by chemical information? Say this hypha is growing on a very extensive area. So, say for example here in the growing region, here in this area, we know that the apical area of any hypha, it is the growing region, and it grows, and this is the this entire area, it in this entire area interacts with the chemical nature of the immediate surrounding. So if there is any food matter, then the hypha will grow in this direction, and if there are certain retardants, the hypha will contract and move in a separate direction. So this information, it is provided from the tip of the hypha to the rest part of the hypha. This information is provided. So the central pore not only helps in maintaining the protoplasmic continuity, but also helps to maintain the transport of nutrient and information in the fungal hypha. So the septation can be also of two types. If it can be just a simple pore, like here what I have shown this in this picture. This can be just a simple pore that is found in the lower fungi phycomycetes and in the higher fungi asco and deuteromycetes. But this complex, there is another type of septation, that is where the pore forms a pore complex. It is a bit more complex than the simple pore. It is found in the basidiomycetes mostly, and it consists of a central pore. It has a septal pore and a thick annular rim-like structure that is called the septal thickening, and this is the septal plate, and the pore is girded by a collar-like structure. As here we see, this collar-like structure is called the parenthosome. So this type of complex pore, it is also called dolipore, dolium, and this type of arrangement is found only in the basidiomycetes. So the structure of the fungal cell. Here we see this is the structure of a unicellular, and this is the structure of a hypha or mycelial fungi. So here we see that every fungi, apart from it has a cell wall, and the cell wall, characteristic feature of the cell wall of fungi is that it will contain chitin. Chitin is present in the cell wall of any fungi, any cell wall we come through, then we can say that this is the cell wall of fungi. So chitin is a characteristic feature of cell wall of fungi. Chitin is present in all type of fungi, but in certain type of fungi, especially in the phycomycetes group, that is the lower fungi, there is one oomycetes, and this oomycetes, which mainly contains the harmful pathogenic fungi, like for example Pyronospora, Pythium, Phytophthora. So these type of fungi which belong to oomycetes, they have cellulose in their cell wall. Apart from chitin and cellulose, glucan and mannan are also present, and this chitin is present in all type of fungi. Cellulose is present only in the oomycetes, and the glucan is again present in all type of fungi. So these are all polymers: chitin, cellulose, glucan, and I forgot to write another is a mannan. So these polymers, they are the fibrillar portion. They form the fibrillar portion of the cell, and they are laid in parallel layers to the plasma membrane. So this is the cell wall, the hyphal wall, and inner to the hyphal wall, is the plasma membrane. Inner to the hyphal wall is the plasma membrane. Membrane composition is similar to that of any plasma membrane, but one thing is that the plasma membrane, it forms folding that invaginate the and goes deeper in the protoplasm. So such type of invaginations, it is called lomasomes. So this type of this is called the lomasomes. Here it is the, here we see is the lomasomes. So the plasma membrane and lomasomes, apart from that, there is a cytoplasm. We see this green structures, and this the cytoplasm, it is composed of endoplasmic reticulum, which is much vesicular, and it contains many mitochondria also. Here we see that every cell has many mitochondria and Golgi apparatus, and of course it will contain vacuoles to store toxic matters, and it will contain inclusions which are also non-living. It contains the stored food matter, that is the glycogen and oil droplets. Glycogen and oil droplets are characteristic assimilatory reserve food matter in the fungal cell. Just like in algal cell we have starch, in fungal cell glycogen and oil droplets are the reserve food matter. It also have a nuclei, and I should mention that this nuclei of fungi, when they divide, division is not typical mitosis, but the nuclear membrane, it remains intact, especially when we see the hyphal form, and it is multinucleate, and if it is dikaryotic, that is two different type of nucleus is present in the hypha, this type of the nuclear division with intact nuclear membrane is helpful in preventing the fusion of different type of nuclei when it is undergoing the process of division. So the nuclear division in fungi, most of the cases, it is with the intact nuclear membrane. So then we come to the flagellar portion. We know that in the lower fungi, that is the phycomycetes, they are mostly aquatic, and they have reproductive spores that contain this flagella. So flagella is absent in the reproductive spores of higher fungi like ascomycetes, basidiomycetes and deuteromycetes or fungi imperfecti. So this flagella are found only in the phycomycetes. Flagellation is very important in the fungi, because this flagellation, whether flagella is present in the fungi or not, also helps in the classification of the fungi. For example, if it is a primitive fungi, lower fungi, then it will have this flagellation, either single or biflagellate, but higher fungi do not have motile reproductive spores or motile reproductive units, and that is why they are considered more advanced and higher. So flagellation, just like any bacterial or algal flagella, we know that in algae also, some of the reproductive units, they have flagella. So just like the flagella of bacteria, algae or this fungi also, the structure is 2 plus 9, that is 11. It has 2 central fibril and 9 peripheral fibril. Here we see this. This is the picture where 2 central fibril and 9 peripheral fibril together, they are surrounded by a cytoplasmic sheath and plasma membrane to form the flagella. Now this is the cross section of a flagella, and flagella are found only in the phycomycetes, absent in the higher fungi. So this number of flagellation can be two types. It can be uniflagellate, that is in the lower fungi, that is the phycomycetes, chytridiomycetes and hypochytridiomycetes. These two lower group of fungi will contain single flagella. In the chytridiomycetes, the flagellation occur in the posterior part and it has a single flagella, which is the example here. And in the hypochytridiomycetes, it has an anteriorly placed flagella. And in case of biflagellate, we have plasmodiophoromycetes and oophoromycetes. So the biflagellate, this is the example, plasmodiophoromycetes, which has two flagella and they are most anteriorly placed. And the oomycetes, they also have two flagella, but they are also anteriorly placed, but the flagella is of two type. Here in plasmodiophoromycetes, here we see that both the flagella are whiplash type, that is plain and simple, and in oomycetes they are tinsel and whiplash type. This is the tinsel. In this is the example of tinsel, where we see that there are two layers of this femur here. So the tinsel flagella, also called the pantonymatic flagella, and this tinsel flagella, they have two layers of this flemurized here, and due to this two layer of flemurized here, they are also, the femur here is also called mastigoneme. The femur, this femur hairs are also called mastigoneme. So this is the example of tinsel. This is the example of whiplash. One thing about whiplash is that whiplash can also be of two types, and depending upon the tip. If the tip of the flagella it does not contain an end piece, then it is called a blunt tip flagella. It is blunt, or it can be pointed end. So if the end piece is present, whiplash flagella is pointed, and if it is not, then it is blunt. Whiplash flagella, it is also called acronematic. I forgot this. This is a wrong spelling. Here it is acronemata, but it will be acronematic. Pantonymatic and acronematic. These are the two types of flagellations. Aggregations or modification of hypha. In the previous slides, I have discussed about how the hypha mycelium is and the internal structure, but there are also certain modification of hypha in especially higher algae. Now, one such modification is the plectenchyma. In plectenchyma, the hypha, it remains attached in such a way that it appears like a tissue. So this tissue-like appearance is due to the fact that the hyphae, they are closely placed, and they give the appearance of a false tissue. And this plectenchyma can be of two types, that is the prosenchyma and the pseudoparenchyma. In the prosenchyma, the hypha, here we see it is the prosenchyma. The hypha, they are arranged parallel to each other, but close to each other, but not so tight that they will lose their identity. That is, here we see we can easily identify each and every hypha. And so they are just loosely arranged, and there is no fusion of the cell wall of the surrounding adjacent hypha. So this type of arrangement is called the prosenchyma. And another type of plectenchyma is the pseudoparenchyma or the paraplectenchyma. In this type of hyphal modification, here the hypha, they are also parallel to each other, but here what happens is that the wall of this adjacent hypha, they fuse, they lose their identity and they form a structure that that look like the tissue. They resemble a tissue but not a true tissue, but each hypha, they lose their, as they are fused, their walls get fused, they lose their individuality. And so this is the cross section. This one is the cross section of a pseudoparenchyma. And from here we can see that it looks just like the parenchyma. And this type of structure, it is mostly found in the fructification of the higher algae. We know that higher algae, it has various type of fructifications, and this type of structure, pseudoparenchyma, is found in the fructification. Another type of aggregation in the fungi are rhizomorph or Sclerotia. This is the picture of rhizomorph, where we see that the hypha, they run parallel to each other, close to each other, and they resemble a root-like structure. So this rhizomorph is also goes by the name of mycelial cord, and they perform the function of assimilation of food. They look like a root-like structure, and this close hyphal association where the hypha run parallel to each other and they run parallel, and this rhizomorph is mostly it is found in the parasitic species, because this structure, it helps in better infection and penetration than a single hypha. So this rhizomorph, this is mostly found in the infecting hypha, and the cell wall of the parallel hypha also anatomos just like the pseudoparenchyma. So this rhizomorph also have the fusion of the cell wall of the adjacent hypha just like the pseudoparenchyma. Next structure is the Sclerotia or the Sclerotium. This is practically a compact, here it is shown, it is a globo structure. It is a circular structure, but it can also be flattened structure. Here the example is given it as a circular structure, but it can also be found as a flattened structure. So these both are the Sclerotia. Basically, Sclerotia is a perinating structure or resting structure. When the amount of moisture in the atmosphere, it goes down, then the cell wall of the hypha, they become thickened, and all the hypha, they form a compact mass, and this compact mass of thick walled hypha, it can perinate for years, say for several years, and on return of favorable condition, they germinate into a new mycelium. So the other modification of hypha or mycelium is the stromata. This is a reproductive unit. Actually, when the sclerotium that we discussed in the last slide, when it germinates, it first forms a stromata-like structure, and then it forms a mycelium. So this stromata, they are very compact mass of hypha, and they are also pseudoparenchymatous, which means that there is fusion of the hypha. In the stromata, the outer layer hypha, they are dark colored, and the inner layer, they are light colored. So this is about the stromata. Then we have Pseudosclerotium. Pseudosclerotium, it is not a true hyphal structure. Actually, when it is formed, it is generally formed at the base of the hyphal reproductive structure, and this actually it is a mass of hypha surrounded by grains of sand. So this pseudo, it looks like a sclerotium, so it's called a pseudosclerotium, but these are just hyphal aggregation. They are not perinating structure, they are not reproductive structure, but just some hypha surrounded by fungal hypha surrounded by particles of sand. So this is the example, this is the picture of pseudosclerotia. Another most important structure of the fungi is the hyphal traps or snares. In the predaceous fungi, this type of hyphal snares, we see there are two types, ring and constricted. So this type of snares, they help in the capture of small animals. For example, the nematodes, the protozoans, they are predated by this type of snares. Here we see, this is the nematode which is ensnared by the fungal hypha, the hyphal snares. Here we see, and usually once ensnared, this nematode won't be able to move, and the fungal hyphae, it will absorb all the nutrients from the nematode. Another modification of the fungal hyphae is the aprasorium. The aprasorium, here in picture we see the aprasorium. It begins, and the fungal spore, here it is the example of uridospore. What it does is that the spore, it grows into a germ tube. Here it is a germ tube. So the tip of the germ tube, it takes environmental cues. That is, it detects the presence of certain chemicals. We know that the plasma membrane or the cell wall of every plant species is different. So any pathogenic fungi, it searches for the specific chemicals that is present in the plasma membrane of its host plant. So it searches for the host plant. So once it gets information about the certain proteins or certain structural elements in the plasma membrane of its host plant, it will detect the host plant by these chemical cues. Once that is detected, the germ tube, it moves in that direction, and once it reaches that plant surface, then the tip of the germ tube, it enlarges and forms an aprasorium like structure. So the aprasorium are only formed when the host plant is present, and they are found in only pathogenic fungi. So this aprasorium is a bulbular structure, and it helps in attachment of the developing germ tube to the surface of the host plant, and once attachment is made, then the germ tube, it further grows inside the tissue of the host plant. So once inside the tissue, the fungal hypha can grow in between the cell, that is the intercellular, or it can grow inside the cell, that is the intracellular. So this aprasorium is basically present in pathogenic fungi and helps in attaching to the surface and also take the chemical cues, and aprasorium development is succeeded by the development of penetration peg or infection peg or infection. Another fungal modification or hyphal modification is that of the Hostoria. As discussed in the previous slide, any fungal spore, it begins its life cycle, it germinates into a germ tube here, develops an apressorium, which is bulb-like structure, and after the apressorium is developed, after it attaches firmly to the plant surface, then it develops infection peg or infection tube and enters the host cell, where it may grow in between the cells, that is intercellularly, or it can grow through the cells, that is inside the cell. If it is growing in between the intercellular space of the cells, then such type of fungal hypha, in order to absorb food, develop this Hostoria-like structure. So Hostoria is found only in the pathogenic fungi, that is fungi which is parasitic on the plants. And so this type of fungi, they, in order to absorb food matter from the organism, that is a plant or animal that it is growing on, it develops this absorbing structure called Hostorium. So basically Hostorium is an absorbing structure. It is developed from the tip of the growing hypha, and it actually here the Hostorial mother cell, it secretes certain chemicals, that is extracellular matrix chemicals, that dissolve the cell wall of the host plant. Here we see that the cell wall of the host plant is dissolved, but not the plasma membrane. So once the cell wall is dissolved, the hypha pushes the plasma membrane and grows inside the cell. Once it is inside the cell, this thin hypha, it enlarges to form a bulbular structure, or it can also form a finger-like projection here, but it will depend upon the species of fungi. Any species of fungi, they can either develop into this type of Hostoria, finger-like, or a bulbular knob-like structure, but never can one species have two type of hypha, two type of Hostoria. So these are the two type of Hostoria, but the function is same, that is absorb nutrient from the plant. And by absorption of nutrient, we mean that the interaction between the hyphal membrane, and here the yellow colored, the plasma, the red color is the hyphal membrane, and the yellow colored, it is the membrane of the, plasma membrane of the host plant. So interaction occur between the host plant and the fungal hypha, and in this way the food absorption occurs. So food is mostly absorbed directly from the cytoplasm by the help of the plasma membrane. So plasma membrane of the host cell as well as the hyphal membrane, they help in conduction of food from inside the host cell to the fungal protoplasm. So this is all about the modification of and with this, I come to the end of this video. The video was all about the structural features, the structure and various modification of fungi, fungal hyphae, and I have also made a video regarding the general characteristic of fungi. I have put the link in the description. Also other fungal related videos, the link has been put in the description. My next video will be about the reproductive feature of fungi in general, and the upcoming videos will be about detail about each of the fungal category, that is for example rhizopas, mucor. So in any case, I have missed any points or how you liked this video. Do write it in the comment section, because your comments will help me to improve my video further. And also, if you want me to make a video in any topic, do write it in the comment section. And if you like it, do share it with your friends, and please do subscribe, because your subscribe will help me, encourage me with this channel. Thank you for watching this video.